All right, ladies and gentlemen, we're going to put all of this protein synthesis stuff together. We're going to start from a piece of DNA and go all the way to a protein. Let's go. So, if the sequence in the DNA molecule calls for a protein with the following codons, these are the questions I want to ask. What would be the sequence of mRNA? What would the sequence on the tRNAs be that match the mRNAs? Complement, I should say. And what would the amino acid sequence of the protein be? So, we need the DNA strand first. So here's our DNA, and then these are the nucleotides divided into their codons. So, first and foremost, we need to figure out what the mRNA would be. Now remember, we're just going to use complementary base pairing rules, but we're making mRNA. So instead of T's, we're going to use U's. A pairs with U, A pairs with U pairs with A, G with C, and C with G. So the mRNA is going to look like this. And before I show you this, pause it now and try to make the mRNA strand using complementary base pairing rules. So AUG, AAU, GUU, UGG, UAU, and UAA. This is the mRNA. This is the complementary sequence to the DNA. This is what's going to leave the nucleus and head out and find the ribosome. Now remember, the tRNA is going to be complementary to the mRNA, right? So it's going to be the opposite of the mRNA. So it's going to be UAC, UUA, CAA, still following complementary base pairing rules. U's pair with A's, A's with U's, G's with C's, and C's with G's. ACC, AUA, and lastly, AUU. Now, remember, we started with DNA, and then mRNA is kind of the opposite, right? And then tRNA is the opposite of mRNA. And if you compare DNA and tRNA, tRNA is the opposite of the opposite. And I know that gets kind of confusing, but if you notice, the tRNA is the exact same sequence as the DNA. It's just you replace the T's with U's because thymine, the T, doesn't exist in RNA. It's uracil instead. And if you use your codon chart, all right, so take a second now, pause this, open up your codon chart, preferably the circular one. That's probably the easiest to read. And try to figure out the amino acid sequence. Remember, we only use the mRNA codons when using the codon chart. Only the mRNA codons, because that's what the ribosome and the tRNA use to bring in those amino acids you're trying to figure out. So the amino acid sequence reads methionine, asparagine, valine, tryptophan, tyrosine, and the last one, and it might be blocked by my camera screen here, is a stop codon, and you should have gotten stop there. Okay, so let's take a more detailed look at translation, this process of the ribosome using mRNA and tRNAs in order to make a protein. So a ribosome is made up of, and we know this already back from Unit 3 when we talked about organelles, 2 subunits. And remember, their names were really long and hard. They were large and small subunits. Just kidding, super easy. Now the subunits are constructed of proteins and RNA. The RNA that helps make up the ribosome is called rRNA, ribosomal RNA. Now these subunits are made in the nucleolus. Remember, that's the whole point of the nucleolus. That more dense region inside the nucleus is to make ribosomes. So when the large and small subunits join to form a functional ribosome, they're going to attach to an mRNA molecule, right? We have the large subunit here. Never mind these green and purple parts just yet. We're going to get to those. We have the large brown subunit here, the small subunit in brown, and then the mRNA, because remember, mRNA is single-stranded, not double-stranded like DNA, sliding right through the middle here. So small subunit, large subunit. Now each ribosome has a binding site for mRNA, and 3 binding sites for tRNAs. That's what these teal colored objects represent. These are tRNAs. Notice the ribosome has 3 places for those and 1 place for the mRNA to go. So mRNA and our tRNAs, delivering amino acids at that, which are these purple shapes. Now, what's really important to notice is that the growing chain of amino acids, right, is held in the middle space where tRNAs can exist and go to in the ribosome. And that's what's going to happen here. This guy is going to move over, this amino acid will bind to this growing chain, and this tRNA will be kicked out like this one just was. And this tRNA will go find another amino acid and bring it back to build more of the protein. Okay, so this is translation in a nutshell. This is what it's all about. So transfer RNA, or tRNA, begins at the AUG, which is the start codon. And it's always AUG. AUG is always the start codon in mRNA. All right, for our understanding. It isn't always. If you go on in later science classes and in your careers one day, you'll find out the differences. But for our understanding, AUG, start codon. Each transfer RNA has an anticodon whose bases are complementary to the codon on the mRNA strand. A tRNA arrives carrying that amino acid that corresponds to the codon on mRNA. Specifically, AUG's amino acid is methionine. Now, another tRNA is then going to arrive with the next amino acid. So, we have this tRNA entering. The anticodon here is going to match the codon or complement the codon on the mRNA. And then we see some ATP, this little explosion emoji thing being used. That represents ATP, energy, right, being used. That's going to create a peptide bond between these amino acids. That's going to jump this chain over to this tRNA. See how it moved from the middle one to the far right one? And then the far right tRNA will move to the center position, bouncing out the tRNA that was once in there. And this tRNA is now free to go get another amino acid. And this is going to continue. This exact cycle that you see here, tRNA is coming in with single amino acids, energy being used to create a peptide bond between amino acids. The tRNA that just entered, moving to the center position and pushing out the tRNA that was in the center position so that tRNA can go find another amino acid. It's going to continue until the mRNA strand reaches a stop codon. Now, amino acids are added one by one until, like I said, the stop codon is reached. The result, once the stop codon is reached and everything's been read appropriately, right? The ribosome hasn't made a mistake, like with the sentence analogy I gave you in the last screencast, then that protein should be functional and go perform a job in the cell or in the organism as a whole. Now, we're going to stop here, and we're going to have a short screencast after this one, folks, to wrap up this unit. Thanks for being here.